So thank you for joining us for this presentation on the KETO-PKD study. I'm Lise Hoover, and I'll be introducing our speaker in just a moment. Before we get started, a few housekeeping. All attendees will remain muted throughout the presentation to ensure good audio quality. We are recording the webinar, and we'll post the recording on our website within the next few days. If you have any questions during the presentation, please type them into the question box located in your control panel on the right of your screen. Okay, this session is designed for informational purposes only. It's not intended as advice on the specific treatment or diagnosis of PKD. And as you listen, if it sparks questions for you regarding your own disease journey, we ask that you please speak with your doctor. All right, so I'm delighted to introduce our speaker, Dr. Roman Ulrich Müller. Dr. Müller is head of the AD-PKD unit at the University Hospital Cologne and has established the AD-PKD registry, which collects data on the management of AD-PKD and has become one of the largest cohorts worldwide. He also leads a basic research group that tackles the molecular mechanisms underlying PKD, employing modern techniques in molecular biology and biochemistry. His special interest is dedicated to renal RNA biology, RNA binding proteins, non-coding RNAs, including the discovery of small non-coding RNAs in PKD. So welcome, Dr. Müller. We are so happy to have you here with us, and I'm going to go ahead and turn that over to you.
Thank you very much, Elise. It's a pleasure to be here today. Thank you so much for the invitation. Now let me quickly move to presentation mode, and I'm glad to see so many people that have joined today to hear a little bit more about what we've been doing regarding dietary interventions in polycystic kidney disease with the KETO-PKD study that was actually made possible by the PKD Foundation and allows us now to look a little bit deeper into this topic in a real clinical trial on dietary interventions. Let me start with a quick slide of conflicts of interest. I may say that none of these have any impact on the currently running clinical trial. And a couple of introductory slides of points that you are probably all aware of: autosomal dominant polycystic kidney disease coming with one of its main problems, which is kidney failure that occurs at the age of about 50 to 60 years in 50% of affected patients. And this is obviously a problem we need to face, and we are trying to solve, and we are trying to find new solutions for. In this context, the key question is, how can disease progression be prevented? And as you know, polycystic kidney disease is not a disorder that is happening acutely and suddenly leads to loss of kidney function, but continues over lifetime, and every year a little bit of kidney function is lost. And the question here is, how can we slow this down or maybe prevent it completely? And in the past, a lot of what could be done was mainly supportive measures. So things like, blood pressure had to be controlled, or salt intake was advised to be low. These are things that are rather general measures that are advised to all of our kidney disease patients. But knowing that there are very specific things going wrong in a kidney in AD-PKD, there's a question whether there couldn't be more targeted therapies that really address the problem that is happening in the biology of the kidney. And in order to talk about this, let me quickly start with a couple of thoughts on how the kidney works and what's the problem in AD-PKD to have everybody on the same page. Now, each kidney of a human being consists of about 1 million filtration units that indeed filter blood and produce urine that we produce every day. And those filtration units are all made up in the same way. And what you see up here is what we call a glomerulus. That's the filter, the actual filter. And this filter filters the blood, and then something we call primary urine, so unprocessed urine, the first filtrate, runs down a tubular system until it is indeed excreted. Now, in ADPKD, cysts form along these tubules and along all of the tubules, all of the tubular segments. And these cysts, what are they? They are mainly fluid-filled balloons, and those fluid-filled balloons are lined by so-called cells, epithelial cells, and I'll also tell you a little bit what these are. What you see here on the right side is actually like a microscopy of one of these tubules. So what you see here is the wall of the tube on the left and the right side, and in the middle of these two walls is where the urine flows. Now these walls are made up by so-called cells. Each of these rectangles is a cell. In the middle is what we call a nucleus that every cell has, is like the brain of a cell, and these structures make up the tubular walls, and this is indeed the actual structures where things go wrong in ADPKD and that start proliferating and start filling, start producing those fluid-filled cysts. And in ADPKD research, what we try to do is we aim to understand what's really going wrong in the biology of these cells. What are the actual things that don't work as they should on the molecular level? And in order to go there, we need to kind of imagine what these cells are. And I already told you there are these rectangular structures here, and in the middle there's the nucleus, like the brain, the structure that organizes everything in a way. And then there are other structures within such a cell. And if you wish, you can compare this in a way to a factory in which different departments have to cooperate in order to make the whole thing work. And this here is a so-called mitochondrion, could be something like the power plant of this factory. And if one of these components doesn't work anymore, the whole thing doesn't work, and that's exactly an aspect that is useful for studying ADPKD. We need to do this in order to find targeted therapeutic strategies to maintain kidney function, which is a key aim in polycystic kidney disease. Now, don't be worried. This is a slide I'm not showing in order to go through all of the things that you see on here, but you may recognize again one of these cells, right? These are rectangular structures, the nucleus in the middle, and then you see a lot of names here, and those are so-called signal transduction pathways. So the molecular pathways in which things do indeed go wrong in polycystic kidney disease. And why or how have we learned this? Now, this is a long process. Decades of research in the field have led to an increasing knowledge of these pathways and how they are changed in ADPKD. And this also is possible because there are model organisms that can be used in the field, and there are some that are somewhat expected, others like worms and flies that maybe are not so much expected to study kidney disease, but all of them have contributed to get towards this understanding. And one of the key aspects, and that's the only one I want to show here today, is a signal transduction pathway that is called vasopressin receptor signaling. And the reason why this is important is because due to this knowledge, due to what we've learned, what's going wrong in the cell, the first targeted therapy for ADPKD could be designed, tested, and approved. And you know this therapy, this is tolvaptan, that has now been approved in many countries worldwide, for example, in the European Union and also in the US to treat ADPKD. What you also see on this slide, and I'm again not going to go through all of these different therapies, compounds, other therapies that have been tested, are currently being tested in clinical trials as active research, but none of them has been approved yet, so none of them can be used on an everyday level. There's also some aspects that have been tried and failed, and this is common in clinical trials. It's not so simple that everything directly from an animal model, something that works there, can be translated to human beings. One of the latest trials there, you may have heard about the so-called staged PKD trial that was examining a very promising compound that was called vanglustat. And this trial was halted because there were no signs of efficacy, and it wouldn't have been warranted to continue and expose people to potential side effects as well. Now, as I said, the only one approved is tolvaptan, which is approved for treatment of rapidly progressing ADPKD based on several clinical trials. But there could also, whenever you talk about a drug, there could also be potential problems, and there could be side effects, and there could be problems as well regarding how how efficacy is in the context of stopping loss of kidney function. And in tolvaptan, one of these problems is that it leads to a strong increase in daily urine volume. So patients on therapy need to go to the bathroom once per hour, 2 to 3 times each night on average. Majority, and I also have to say this, of our patients still considers the therapy feasible in everyday life and does continue in the longer term, but this is a challenge, and there are also other side effects. And regarding efficacy, it slows down kidney function loss, and this is a great thing. So this was really a milestone for care in ADPKD, but it does not stop it. So it does not essentially prevent the need for renal replacement therapy, dialysis, kidney transplantation in the longer term. And this leads to the conclusion that whilst it is a milestone, that there is a first targeted therapy, we need other future options. And the question is, what are the most promising future options? And I would like to share one aspect on this today. When we think again about what's going wrong in these cells, so in the walls of the tubules in the kidney and the walls of the cysts, one key term here, one key word is metabolism. And I'd like to show you why this is the case. Whenever we do studies in ADPKD, we find that one of the key things that is changed is cellular metabolism. So what does this mean? Now, this is a very simple definition that's actually derived from Wikipedia, where you can all read this, but it's an important definition of metabolism. What are the main purposes of metabolism? So metabolism is the conversion of the energy that is in food that all of us eat every day to energy that's available to these cells and that they need to work normally. It's also the conversion of food to building blocks. So building blocks that are needed to build up those little factories, the cells. But there's one term that's in both definitions, and that is food. And that also leads to the question, can we do something about the cyst formation? And this is a drawing from the 18th century already of a cystic kidney by changing our dietary habits, which would be quite amazing because that's something everybody could do on their own in a way. And we know that dietary interventions in general can be quite powerful. I already told you that there are animal models for ADPKD, so people in the field wondered, why don't we test whether food intake indeed has an impact on disease progression? And this may be a little bit hard to interpret what you're seeing here first, but this is a cross-section through a kidney, and you see a healthy animal. That's how a normal kidney looks, and you see PKD, and you see what you would expect, a lot of large cysts everywhere. But if those animals underwent a diet, so just 23% less calories, the cyst formation was greatly ameliorated. And that was obviously a first indication that changes in dietary habits could have a role in ADPKD. But it also raised another question. Is it really all just about the amount of food? You know, calorie restriction means you just take out a bit of the food, you eat less. Is this really the mechanism, or is it really required for the effect, or is something else happening in, again, the metabolism, so in the way the cell uses food to create energy and building blocks of a mouse on such a diet? And this is a very important thought, and that's a thought that is underlying a lot of what we're doing right now at the moment in research. If mice get less food, they eat everything at once. You know, they would eat this once a day, and the rest of the day there would be nothing left. And when there's nothing left, well, what's the consequence? Time-restricted feeding, intermittent fasting. You know, they would feed for a couple of hours and then be fasting for the rest of the day. This is something a lot of us know as the 16+8 schemes, you know, 16 hours fasting, 8 hours eating, that are discussed a lot at the moment, whether they're healthy and how they can be used. What does the body do, and what does the cell do in such an instance when there is fasting, when there is no more food supplied? Now, the body starts burning fat instead of sugar. That's a general thing that always occurs in these cases. And this leads to the formation of so-called ketone bodies, that are products that are formed during metabolism of fat. And that is what we call ketogenesis. and is the key concept behind ketogenic diets. And this is a concept you can again test. Does this ketogenesis that occurs in mice when they are on caloric restriction indeed lead to the beneficial effects in polycystic kidney disease? And this is something that was done in a very, very nice study by Jacob Torres and Thomas Wimes from Santa Barbara, whom you may know, in which they tested, and was published in Cell Metabolism in 2019, whether mice that got a normal amount of calories would benefit from a so-called ketogenic diet that causes the formation of these so-called ketone bodies. And this was indeed the case, and that's just shown in this drawing here on a high carb, so a lot of carbonates, as we all eat, or most of us every day, a lot of glucose available, form polycystic kidneys. Now, if you give the same strain of mice a ketogenic diet, less carbohydrates and a lot of fat in the end, this cyst formation is blocked and much ameliorated. And that was an amazing finding because this proved to be one of the most powerful tools that had ever been examined in any of the polycystic kidney disease models. It also raises the question again, how can we as humans potentially reach a ketogenic state if this may be beneficial in the future? And there we again we have to talk about the time restricted diet, so eating a certain number of hours, maybe 8, maybe less hours a day, and being fasting the rest of the day. That's a potential way to get there. Humans are a little bit more resistant to ketogenesis if this is the strategy than mice would be. Intermittent fasting, and this is sometimes a little bit confusing. There's similar terms I use, but the actual intermittent fasting is something else. So you do fasting for a couple of days, a week, a month, and then the rest of the days you eat normally. That's what really intermittent fasting is. That's a way to reach a ketogenic state. And the last way is the classical so-called ketogenic diet. And what does this mean? I already told you before, it's a lot about burning fat. So you would increase fat intake, would go along with a massive reduction of sugars, carbohydrate intake around 20 grams per day with a normal protein intake. And if you do this, and there we're back to the animal model data from the Wimes lab, and then compare the normal diet and the normal diet carbohydrate, which is shown in black here for males and females, and the ketogenic diet, so this diet rich in fat and low in carbohydrates, are compared here, that's shown in red. And what you see here is the percent of cystic area per kidney, so how much is the cyst load, and you see that this is much ameliorated by a ketogenic diet. So this is a very powerful effect. And obviously, and that is clear, if you see something so powerful and you're looking for new therapies, also raises the question, should all patients now follow a ketogenic diet? This is really powerful and obviously very attractive. Now, the answer is a little bit more complicated and probably has to be no by now because data in humans are missing. And this is a problem, and I'll also share with you why this is a problem if we have no data in humans. I've already indicated that before when I talked about therapies that were tested in clinical trials and were very, very potent in animal models, but never made it to approval. And why? Because it did not work in humans or there were safety concerns. Now, I'm still willing when individual patients come and say, no, I want to try this now, I want to do this because it's not a drug and it's a dietary intervention. It's just a change of dietary habit in a way. And if people eat in a conscious manner, that's very often better than not looking after what you eat every day at all and you just eat random things. I would tell them what's a potentially healthy way of reaching a ketogenic state, but I would not advise anybody to do this on a general level now. I mean, it's clear that this will not be the right way. This is a way to reach a ketogenic state, but if you only eat things like this has other negative health impacts. And there are ways that are mainly vegetable based, and there can be other food sources like salmon and so on. If you go for a healthy ketogenic diet, that would be, if you go for a ketogenic diet, the right way to do this. Nonetheless, there's no general recommendation at the moment from the medical perspective. And this is due to the fact that we have to ask whether ketogenic diets are safe and effective in humans at all. And there are reasons to believe that they are safe. Many people follow ketogenic diets for different motives. People want to lose weight, people believe this is healthy, and there are no major safety signals that we observe yet, but there are a couple that have to be considered. Ketogenic diets have already been established in medicine for severe epilepsy in children and are potent there. So this is something that can be used. This is a good indication of safety, but it's not a proof, and especially not a proof in certain populations that suffer from specific diseases like kidney disease. Scientific data that are available describe a row of positive effects, but yes, there's nothing known yet regarding ADPKD that has been examined in a structured manner, and that's why we're a little bit careful there. The consequence is clinical trials are urgently required to look at efficacy and to look at safety, but also look at the fact whether this can be done on a daily level at all, or whether this is only feasible for 5% of patients and then may not play this major role either. And this is exactly the questions we're asking with the KETO-PKD trial that is funded by the PKD Foundation. What do we do there? We examine two different ketogenic dietary interventions over a period of 3 months. 63 participants is what we're aiming for. And we mainly ask for the question feasibility. So can this be done on a daily level? Are there problems? What's the best way to do it? We're asking for safety signals, and as a secondary endpoint also efficacy. So we look at total kidney volume. You may know that the size of the kidneys and you have to look at this in an age-adapted fashion, but this tells something about future kidney function loss, and usually slower growth of kidneys is also associated with slower loss of kidney function. Now, let me quickly guide you through the trial design. We screened patients that took part in our registry, and this is exactly why those cohorts and registries worldwide are very important. That's one of the reasons. It's not only to characterize the disease and learn more about the disease, but also to be in touch with patients that can then be potential participants in trials when we ask them. We aim to screen around 160 patients in order to find the required number of participants. This was enabled by the fact that in the registry there are around 1,000 patients now that we could get in touch with. Now, after this has been done, patients have to be enrolled and have to be randomized, and this is something we're doing with 63 patients. What does randomization mean? We don't want a bias. If patients or we decide who is in what treatment, which diet, then this is a bias, The reason already, there may be certain reasons why certain people, let's say all young women in the trial, choose to be in the ketogenic diet arm, and all older men choose to be in the control arm, and that makes it impossible to compare the results later. So this has to be done in a random fashion, and we did this and stratify by severity of kidney disease and gender. Randomization is done for 3 arms, as mentioned previously. Ketogenic diet, so high in fat, low in carbohydrate, is one arm. The control arm, those people are told what's generally considered a healthy diet in kidney disease, but they go on eating a carbohydrate-rich diet as before. And the third arm, again, a ketogenic dietary intervention, as I told you before, is water fasting. What do these participants do? They, for 3 days, once per month, do not eat anything. They drink a lot of water. They can have a cup of broth per day, but they should not have any carbohydrate intake and should not eat anything else than this. And that's just 3 days a month to make it feasible. Now, the intervention itself, the diet, goes on for 3 months, and then at the end, and that's aimed for after close to 2 years of running this trial, is what we're really looking forward to, data analysis for, is this feasible? Does this have an impact? Does it have an effect on kidney volume? And is this safe? And what does this mean for the individual participant that takes part in our trial? That's 6 visits at our hospital. So they have to come 6 times. And we're very happy that so many people are interested in doing this, because this really helps to learn for the whole community. There's a so-called preparation phase. 14 to 28 days, in which we talk about everything and prepare the participants. And then we do a first MRI to measure kidney size, kidney volume. A couple of other visits during the dietary intervention, during which we measure a lot of things like blood values, and then there is a second MRI after the diet to compare the kidney volume, and the period that in clinical trials is always called washout. What does this mean? Well, this is you go back to your normal diet, or you stop the drug that was examined in a trial for a month, and then we do another MRI at the end to see whether there is, if this works here and kidneys get smaller, is there a rebound? Do they get larger again? What's the effect of going back to a normal diet? And that's the overview of the trial protocol. What's the current status of the trial? Now, I'm very happy there because this works very efficiently and everybody helps participants, our doctors here, collaborators, our nutritionists, etc. And this is quite a challenge due to COVID, I have to say. This complicated things a lot, but this works very nicely. 24 patients have completed all of these visits by now. 28 are active somewhere in the trial, and 11 have an appointment to start. And you may recognize that this is the 63 participants we're aiming for. The last patient is now scheduled to start in January 2022, and the data are then expected, because it takes 5 months to go through the entire trial, in summer 2022. So we'll be able to present first data on this in fall next year. Now, what's clear as well, after this follow-up, we still need to learn more. This will be very nice to have a first insight, but if we want to look at how good the impact of this dietary intervention is on kidney function loss, so what we call eGFR decline or kidney size increase, we need larger multi-center trials that follow up on this, so that involve patients from many centers, and that's something that will be a target, a goal after our trial. Our trial will help already to say which of these diets is the most promising one, right? So be able to probably only examine one dietary intervention. This is quite a challenge. Those large trials require a lot of funding, require a lot of organization. That's all things that still need to be solved in the future. Now, having said this, and also having shared with where we are at the moment, and also the fact that obviously in the middle of a running trial, we cannot share results yet. This also raises the question, can we already learn something from other studies that have been completed, that are currently running, that have been analyzed? And that's the last part of my presentation today that I'd like to share with you. Now, we here in Cologne have done another study that is a very small longitudinal pilot study called Reset PKD that was done with the same, and you already recognize those dietary interventions that were done in KETO-PKD, 3-day water fasting, ketogenic diet, but in this case, just for 2 weeks. So a very short period with the question, does this already have an impact on kidney volume? Because there were indications from the CAT model that was examined before that this may indeed be the case. And also with the question, was this feasible in this short term? Again, and you already know this kind of a design now with measuring kidney volume in the phase that people still eat normal, then before and after the ketogenic intervention, and then again after this so-called washout phase. This was only 5 participants in each arm to learn a little bit here and examine the short-term effects. The study has been completed. The results haven't been published yet. I want to share just some key points with you today on this, so that we see what we can learn from this first pilot study. What I can tell is that both water fasting and ketogenic diet were well feasible. How do we know this in the short term? So 80 to 90%, depending on how you measure, were able to go through the diets without reporting any problems at all, and also reached an endpoint in blood values. So we can take blood samples and measure those ketone bodies and see, does the diet really work? So did the participant stick to the diet, and does it lead to the changes in cellular metabolism that we expect? We can measure that in blood values. And if you look at both, so what people reported and the blood marker measurements, we can say that it was indeed well feasible. There were no relevant safety signals in this short period, and as I already indicated here, very efficient formation of ketone bodies, so that's a good thing, but it did not have an impact on total kidney volume yet in this very short period, so we can't say anything about efficacy. But it's also clear this was only 10 people taking part, a very short-term treatment, so larger trials are urgently needed, and the first step towards these larger trials is now the running KETO-PKD trial. Any other data from other groups available yet? You may know the efforts from Colorado, from Kristen Novak's group that mainly examines obese or overweight adults with ADPKD and did a study looking at intermittent fasting, so 3 days per week, or daily caloric restriction to look at weight loss feasibility, but also change in total kidney volume. She has already, I think, in a previous presentation, shared some data on this, and here we will soon, from a publication, as I heard, learn more about the results, how this works in obese, overweight adults, different protocols that are not necessarily ketogenic, but also have a lot to do with the dietary intervention. And the same group is running another 2 studies at the moment, all in overweight, obese patients, because the aim there is body weight loss with different dietary interventions. And I'm very much looking forward to learning from these trials in parallel. So whenever such things are done, they're not done in redundancy, but they're always done with different characteristics, and we can learn a lot from each other. And thankfully, there is a lot of international collaboration in the field. There's another one, a trial that has been prepared in Italy, no data available yet, but that also looks at a low carbohydrate diet, the so-called modified Atkins diet, compared to balanced normal caloric diet, again looking at feasibility, safety, and total kidney volume. And again, we hope to learn a little bit more together from this trial. So you see, this is an extremely active field. Researchers are really trying to solve this question in collaboration with patients and patient advocacy groups, and we hope we'll be able to do this. Now, when we talk about existing data, we can obviously not only talk about the classical clinical trials. Those are absolutely needed for the data quality we need before we can give advice in the everyday clinical setting. But we can already learn something from real life experiences. And you may know there are people with ADPKD or without ADPKD, but also ADPKD patients that have chosen to follow ketogenic diets for different motives, and those people can be asked about their experiences, whether there were medical problems, how they feel while changing their diet to a ketogenic dietary intervention. And this is something we do within our what we call a translational project pipeline for ketogenic dietary interventions in ADPKD that we run in Cologne in collaboration with the WIMS laboratory in Santa Barbara. And Reset and Keto ADPKD here on the right are the things we're still waiting for. But we can, of course, already learn from real life experiences. And this is what we did in what we call a clinical case series study. In this collaboration, together with the German patient advocacy group, in which 131 ADPKD patients that had already used ketogenic dietary interventions were asked questions about well-being, feasibility, potential side effects. And you may have heard about this before from Sebastian Strubel from Thomas Wimes' lab. He's actually a medical doctor who received parts of his training here in Cologne in Germany, then moved over to do basic research in Thomas' lab, and will again join our group here sometime next year, and who's done a major effort here to perform this study. Now, who took part? As you see here, there were the majority of participants underwent a classical ketogenic diet. Some reported a time-restricted diet, and only very few said they underwent classical caloric restrictions, so just reduced calories on an everyday basis. And I can't present the entire study and all the data to you today. I just want to share a bit of an insight, but this is something you can find in a published manner by the Wimes lab and with our contribution online. If you ask, do you feel that your diet is easy to do for people with ADPKD, the feasibility aspect we also have in our trials, the majority agrees, but you also see this won't be a solution for everybody. So somebody who strongly disagrees this is feasible or somewhat disagrees may not do this in the longer term. This can probably be solved partly by support. So supporting people by dietary advice will help increasing adherence and tolerability. What impact did your diet have on your health and well-being? And this is all self-reported, so no objective measurements like in a trial, but still very important. And you can see that the vast majority said that the health and well-being improved whilst undergoing one of these dietary interventions. A lot of them actually even said strongly improved. When we're talking about ADPKD related symptoms, do you feel that your diet improved your ADPKD symptoms? Again, a significant proportion said strongly agree, somewhat agree. It's clear that this won't be the fact for everybody. You again can see that this all has a tendency towards the positive side. So it's encouraging for us to continue what we're doing, but still the clinical trial data is urgently required, and we really hope we'll be able to solve some of the questions, the remaining questions within the next 1 to 2 years, leading then to maybe other trials, but putting us in the position that we can already answer many more questions that we cannot answer yet. Now, and I've indicated that before, the final answer to the question how much efficacy do we see to treat the disease and to stop kidney function loss? This is always something that will take a little bit longer and will take another larger trial after keto-ADPKD, but we will be able to give a first insight and will surely be able to answer most of the questions on feasibility on what's the right diet for a follow-up trial or in everyday life. And that's about it, what I wanted to share today. This is obviously a collaborative effort here in Cologne with many people from basic research, imaging, cell biology that contribute to what we do in research. That's also extended to the pediatric nephrology, close collaborations. Beside our registry in adults, there is a pediatric registry as well to learn early on how ADPKD, what impact ADPKD has in the life of children. And I'd like to finish thanking some of the people in our team here, Franziska Grundmann, who leads the ADPKD unit together with me, Simon Oehm and Konstantin Steinke, who ran the small pilot trial, the RESET PKD trial, Christoph Lindemann and Sadria Czukowski, who now as our study physicians run the KETO-ADPKD trial, and obviously Sebastian and Thomas for all of the collaboration over the last years. And Thomas together with me designed the KETO-ADPKD trial and helps us a lot with advice. And we've gone through they have not only done the basic research behind all of this, but also things like the clinical case series with experiences from the real life setting that I've just shared with you. And now I'm happy that we could share this, and I'll be happy to answer your questions that you may have.
All right, excellent. Thank you so much. That was fantastic. I believe, Shauna, can you see both of us on the screen for answering questions?
Yes, I can.
Excellent, great. All right, so we had a lot of great questions come in. We had a couple about keto in general and research, and then a couple about the studies. So let's start. Do you have any information about how keto might impact an AR PKD patient or animal model?
Yeah, this is indeed a very important question, and is something that's being looked at in research now for patients. I mean, as I said before, we don't really know yet for ADPKD, even an AR-PKD is much rarer, and there are no trials running yet, so we can't say anything there yet. The animal work is being started, and that's things we don't have data on yet, but that will be answered. The problem there is a little bit that we have quite good ADPKD models that resemble human disease, this is much harder for ARPKD. So answering these questions won't be so simple for ARPKD. Nonetheless, this is active work and this will be solved, but will take a little bit longer.
Absolutely. What about the keto diet in various kinds of mutations in PKD? So we know there's mutations in PKD1 or PKD2, also various different kinds of mutations in those genes. So do we have any information about how that might impact the intervention?
So this is of course clear. There are mutations in several genes, mainly PKD1 and PKD2, and there are changes in many positions in these genes, right? And this could have an impact on what we see. Now, we do not have any data for patients again. And I believe the strongest effects there we'll see if we see effects in patients that are most severely affected. And that is PKD1, the so-called truncating, the most severe changes in the PKD1 gene. If you think about what comes from the animal models, we can clearly say that the changes in cellular metabolism we see and that underlie the hypothesis why we believe these things work are quite similar in the different mutations. So the hypothesis would be this will work in all and has the greatest effect in the most severely affected patients. When you think about the animal models, we can clearly say that the different rodent and feline models that were used in the studies to date from the Wimes lab have mutations in different genes. And this does not impact whether the diet is efficient or not. So from what we know now, we would say it does not depend on the genotype.
Okay, fantastic. We have another question about the level of ketosis or ketones produced versus just being in a state of ketosis. Do we know if there's an optimal level or if it's just simply being in a state of ketosis that seems to have impacted those animals?
Yeah, this is difficult to say and especially difficult to translate to humans. So there we'll have to see what we reach in humans and whether we see efficacy with the levels we reach in humans. We're right now looking for this is part of the trials that there is a ketone body threshold. So we want efficient ketosis, but we don't want severe ketosis either. And if this may not be something everybody knows what it means, but we're looking for ketone body levels, above 1.0 millimole per liter. So this is something you can measure that's feasible without any symptoms. Do our participants reach that easily? I have to say, with the dietary regimens they follow, they reach very potent ketosis. So we'll be able to tell whether this potent ketosis has an impact or not. And then titrating that down, how much you really need and so on. That's another challenge. That's not so simple, right? I mean, if you want to do that, you need many, many arms in a trial and so on. That can be a topic for future studies. At the moment, it appears that at least, and that's good, potent ketosis is feasible and we'll be able by that to answer the question whether ketosis has an effect at all or not. If we in the first clinical trial had gone for very low level of ketone bodies, we would afterwards probably have said, well, we didn't reach the effect because we didn't have enough ketosis. So this wouldn't have answered the question. And that's why we're rather satisfied that we do think we're seeing potent ketosis.
Okay, great. What about other cysts aside from the kidney? Do we know anything about how it might impact liver cysts, for example?
Yeah, we don't. But again, this is measured in the clinical trial. So we do not only measure kidney volume, we also measure liver volume. So we'll be able to say something about this. Do we expect that there will be an effect? Yes, because whilst many things may be different, you may know that tolvaptan as the only approved treatment does not have an impact on the liver. It has an impact on the kidney. And the reason is that its target where it binds to, only exists in the kidney and is at least only relevant in the kidney as far as we know right now. So that kind of treatments will only work in a specific organ. But the changes in metabolism are quite similar in different cyst types. So we'd expect if it works in the kidney in humans, probably works in the liver as well. Remains to be proven, but we're measuring this too.
Okay. Yeah, well, speaking of the study, so we had some questions about tolvaptan and those who want to volunteer to be a study like this. Could you tell us about the logic of excluding those on tolvaptan from an intervention trial like yours?
Yeah, at least for the first step, we needed that because if the question is always, does the trial answer your question in the end? And this is only possible if you don't have too many confounders. So other factors that also influence the results. And if tolvaptan, is another, and tolvaptan would be a potent other factor that influences the results. And then you can try to again level out for this by putting as many tolvaptan patients in one arm as in the other and so on, but that makes it very complex and is sometimes not possible in a trial of this size if you have to randomize, to put random people into each arm based on kidney function, gender, drug 1, drug 2. This is not feasible anymore down the road, and we felt we now have to answer the question first, and answering the question requires, unfortunately, not including patients on tolvaptan for now. Down the road, the question will be, is the impact of the diet similar to tolvaptan? Is it larger than tolvaptan, etc.? And based on these findings, and we're still waiting for the first proof that it does work at all, right? But based on these findings, one would have to decide, is it an add-on to tolvaptan? Does it replace an existing therapy? Or is another trial required that compares tolvaptan plus diet with diet only, and with tolvaptan only, and to see whether it has an added benefit. But that's again a little bit down the road and would have required, if you see, we're looking at 63 patients, classical drug trial that leads to approval rather includes something like 1,000 to 5,000 patients. So that would have required much larger numbers, and obviously due to that also a huge amount of funding, which wouldn't have been the first step into the field, but will rather be the second and third.
Absolutely. All right, and so in the study, you mentioned there are 3 things you're looking at, and one of them was safety. So what are you looking for when you're thinking about safety in that study?
Yeah, so in general, we measure the key lab values, so the liver panel, kidney panel, lipids, cholesterol, uric acid, and many more. And anything that pops out as something of concern, an increase in liver values and so on, would be a safety signal. Then we also ask our patients, participants about symptoms. So if somebody suddenly comes back and I've been on this diet now and I have flank pain all the time, that would be a safety signal, right? Anything that leads to hospitalization, if there are more infections suddenly, would be checked for. And then there are things like kidney stones, for example, which would be a key thing that can be influenced by dietary habits. That's something we'll measure. But then in the end, we'll see is there any safety signal out of all of this, and is it positive for our diets, or is it negative for our diets, and will it need some pondering? What's the benefit over the risk that you would expose someone to when starting such a dietary regimen? And as you know, I mean, for any drug, there is a side effect. So this always has to be taken into account, the benefit risk ratio. And potentially this is also the case for dietary regimens. Something that has a very potent positive effect can potentially also have negative effects. And that's one of the reasons why we're convinced we need clinical trials in order to rule those out before providing general advice that people should start such a diet.
Yeah, fantastic. All right, how about that water fasting arm? Why not just have the keto diet and the control? Why the third intervention there?
Yep. The reason is we know, there are many different ketogenic dietary interventions. So diets that lead to a ketogenesis formation of ketone bodies, and those are 2 of which that had been examined in the animal models. So, and that's one of the reasons, right? We want to translate those. If we knew beforehand that one of them is safer, better, etc., in humans, we would have gone for this one. We just don't know at the moment. And if we think about a future large trial, let's say 1,000 participants from many centers, you'd rather focus on one diet then, but learn from these first trials, which is the one that's more promising. And that's exactly what we're trying to learn. So right now we don't know, when you think about feasibility, is it more feasible for people and easier to do 3 days water fasting once a month, which can be a challenge, or is it better and easier to undergo a ketogenic diet, but every day, which again is a challenge, at least to some people, right? I mean, this can have an impact on social life as well, because you can't just easily go to a restaurant and say, I eat whatever I get. You have to be aware of what you get. Not everybody offers ketogenic meals, but most restaurants wouldn't. Many people don't if you're invited and so on. So that's exactly why we want to find out now which one of the two appears more feasible, safer, and maybe more efficient as well, to learn from these first pilot trials.
Great. Yeah, we had a lot of people also interested in participating. Could you remind us, what your enrollment looks like right now, and then when you think you'll have results?
Yeah, so the enrollment has been pretty much completed. Which may be disappointing to people that's still looking for participation, but is obviously good for the trial and good for all of us learning together that we are efficiently enrolling. Last patient this means we have gone through the screening and patients have agreed to participate, but not everybody is in the trial at the same time. This wouldn't work. We can't do 63 MRIs on the same day and so on. That doesn't work. So this is done in a consecutive manner, and the last patient will start in January. The whole trial period with the preparation phase, 3 months intervention, 1 month washout takes 5 months. So data will be available in May, June next year. And then you have to reckon we'll need a bit of time to analyze and prepare this in a way that's quite a lot of work to do this in a good and reliable manner that we'll be able to talk about this probably in fall next year. And as you know, one of the large international conferences is always in fall every year. That may be one of the places where we can talk about this these data the first time.
Great. All right, we have time for one last question. This has been so fantastic. Thank you. A lot of questions about what this is going to look like for patients in the clinic. So, once, any kind of research study results are published, as a clinician, what do you think about how long it takes for those research results to reach, a nephrologist? What's the role of the patient to advocate for themselves or to do their own research in these kind of studies?
Yeah, that's always a bit of a difficult question, especially for dietary interventions, right? That, I mean, we as medical doctors, we're quite used to dealing with drugs and new pharmacological treatment strategies. We sometimes have a hard time to think about alternative options. I would think we'll need to come up with new infrastructures there as well, in a way, as to dietary counseling, as to coming towards a dietary counseling that allows for a science-based approach, because there's always the risk when there is these data out, everybody's trying to find their solution, and suddenly many people start offering solutions. But the question is, what is really based on the science, and how can we really offer that to patients in the future? That's things that several people in the field, including Thomas Wimes and me, are thinking about a lot at the moment, and trying to come up with ideas on how to solve that for the future. And we'll try to flank such trials also with future options, to provide advice to patients. What's the right way to do that, and what's the things to keep in mind, and what may be components that people offer as part of a dietary intervention that are not science-based, and others that are science-based, and so on, in order to transport this information and provide patient education and also physician education in a way.
Yeah. Well, thank you so much, Dr. Mueller. This has been fantastic. Great presentation. I just wanted to say to everyone attending, thank you so much for joining us. And like we were discussing, these results from research are not possible without patients like you participating in research. So thank you to those who've been in a study or thinking about being in a study. The foundation has a clinicalstudies.org webpage where you can see what PKD studies are enrolling if you're interested. And you can also email us at research@pkdcure.org. So as a reminder, this webinar has been recorded. It'll be posted on our webpage within a few days, and you'll get a notification of that through your email. Feel free to share with anyone that could be interested. And if you have any further questions, please go ahead and email us. And sorry to anyone we couldn't get to. So many great questions came in. Yeah, thank you for joining us. Thank you, Dr. Mueller. Enjoy your days.
Thank you so much. Bye-bye.
Bye-bye.